Hello. It's good to see everybody. Hopefully we will be starting in just a minute. We'll just wait and see if there's others to join us. Hello, Malaysia. Oh, brilliant. It's nice to see everybody chatting. You're so excited. You've been waiting since yesterday. Hello. Hi, Zara. Hi, Daniela. Good to see you. So we're ready to do a web class today on synapses and reflexes. Hello. So where's everybody from? Be nice to see where everybody's from. Hello. Geeta. We're all good. Thank you, Zara, I think. Ready for a lovely class on a Sunday. Yeah, on GCSE. So it's GCSE synapses and reflexes. Yeah. Would have been great last week. Hi. From Malta. Ah, fantastic. I hope it's a bit warmer over there than it is here. But Malta, that's an interesting one. So I think that we will be ready to get going. So let's have a look at the starting of this. So let me share my screen with you. Hopefully this will come through in just a second. All right. So hopefully you can all see that. Can you all see that okay? From the Maldives, that's fantastic. Sri Lanka, Milton Keynes, yay. Good shout out for Milton Keynes. Brilliant. Yep, brilliant. Let's get going then, shall we? So let me introduce myself first. Some of you haven't come across me yet. I'm the lead GCSE biology teacher. I've got a BSc in animal science from Leeds University and a PGCE in biology and games also from University of Leeds. I'm a teacher and head of biology with 25 years of experience, which I hope I'll be able to pass on to you. And I hope you'll find that I'm really enthusiastic about my subjects. So I've also been an exam marker and I have written for Zigzag Education and CGP, which I'm sure a lot of you know. So what I want to tell you about first of all is the fact that we have got a 1 week free trial so that you can try out and see what this is like. So when you if you like to have a go, and I think you probably would like to have a go, this is if you put in tutor 7 on the Snap Revise website, then we can have a look and you can go on and there's tutoring on there and you can create an account and it's got all the GCSE and A-level subjects that you could want. And I'll go through it with you at the end of this revision session. So I will show you the website and I will show you how to get onto that tutoring at the end. So do have a look at snaprevise.co.uk, put in your tutor 7, sign up and just have a look and see what you think. It's a great deal. You get a lot for your money and it should be really useful for you all to catch up with your topics that you might be struggling with. Okay, so let's get on with the lesson then today, shall we? So let's have a look. What are our objectives today? Well, I want to explain how the function of a synapse relates to its function in communication between neurons. Describe the structure and function of a reflex arc. Describe the importance of reflexes and the investigation of reaction times. What I won't be covering today is I won't be covering the brain or the eyes and vision. Hello, we've got lots of people joining us from all over the world. This is fantastic, brilliant. A great opportunity for me to help you on this particular topic today. Now, do ask questions as we go along, and I will answer them, and I will be asking you questions. So I do want you to try to have a go at answering some of the points that I'll be asking you today. So let's have a quick look at where the specification points come in. So with there's AQA, it comes under the human nervous system, structure and function. With OCR, it comes under describing the structure of the nervous system and explain how the structure of the reflex arc is related to its function. And with EdExcel, it's looking at the structure and function of the sensory receptors and neurons, basically the whole structure of the nervous system and how the reflex arc works. Okay. So what do we already know about the structure of the nervous system from a stimulus to its response? What is the passage along the nervous system from the stimulus? So we start with the stimulus and we go on all the way through until we get our response. Can we come up with what order we go in? And if you've got any idea, so we start with the stimulus. Starting with the stimulus, what comes after? Now, I always teach my pupils to think of it as S-R-S-R-M-E-R. And if you've been in any of my web classes so far, you will have heard me talk about this before. And it's something that people manage to get into their heads, S-R-S-R-M-E-R. And that will help you work out what order we need to go in. So has anybody got any idea? After the stimulus, sensory, yes, brilliant, good. So stimulus is the first S, then receptor, then the sensory neuron, brilliant. So we've got stimulus, receptor, e.g. a thermoreceptor, which detects temperature. Sensory neuron. So we've done stimulus, receptor, sensory neuron. Yay, relay neuron. In the central nervous system, we often have the relay neuron, don't we? Relay neuron. Then, so that's the relay neuron. And then we go, yes, brilliant, Neha, motor neuron. Which is the M. Then we go on to, what's the E? What carries out the response? Anybody got any idea? Brilliant. Yes, you're all coming up really well with the answers here. And what carries out the response? Can anybody remember what carries out the response? It's the E. So we're going to end up with the response, aren't we? Response down here. Brilliant. The effector. The effector which brings about the response. So maybe the thermoreceptors have picked up the temperature is increasing outside. So it's passed it back to the central nervous system, which then sends impulses to the effector, which could be, say, a sweat gland, and it carries out the response of sweating. So an effector is a muscle or a gland, and that brings about the response. Well done, excellent, good start. So now we need to look at the different neurons. So can you name and describe the structures of these three types of neurons? So we've just gone through SR-SR-MER, haven't we? So we've gone through SR-SR-MER. So now we need to look at the types of neurons. So what have we got? That first one is, we've already had, was the sensory neuron, wasn't it? Then we go into the center one, which passes the information from the sensory neuron to another neuron, which carries the information to the effector, which is over here. So it goes sensory neuron. What was that middle one? Relay. Brilliant. Well done, Zara. And the motor. So those are the 3 types of neuron. And it's useful to know the differences between them. So if you can have a look, first of all, at the cell bodies, if you can see with the sensory neuron, this is the cell body here. The cell body is the bit which has got the nucleus in it. So the sensory neuron, the cell body is about 1/3 of the way along. With the relay neuron, it's in the middle, and at the motor neuron, it's at the end next to the central nervous system. All right. Now, what is the name of the gap between the two different neurons? So what's the name of that gap where the message is passed on from one neuron to the next? Brilliant. Excellent. So you've got synapses here and here. All right. And does anybody know what this part is called? The bit that's carrying along. Brilliant. So we've all got the synapses of the gaps. And you're thinking about the nodes of Ranvier probably, because it looks like I'm pointing at the gap, doesn't it? But yes, it's the axon which carries the electrical impulse along the neuron from the cell body. It goes from the cell body along to the next neuron or to the effector. And then you have these little gaps. This is obviously a fatty sheath, isn't it? The myelin sheath, fatty sheath or myelin sheath. And there's gaps in between, aren't there, here, which are called nodes or nodes of Ranvier or internodes. So yes, we have little gaps there as well, which are called nodes of Ranvier. But the fatty sheath is there to basically stop the electrical impulse from going sideways, because we want the electrical impulse just to go in one direction. All right, and we just want it to keep on flowing in that direction. We don't want it to end up getting moved across other neurons. It's a bit like your electrical wires that you have, and there's plastic around the outside to insulate them, to make sure that the electricity just goes in one direction and it doesn't electrocute you. Same idea with the axon. It's insulated with this fatty sheath, myelin sheath. Excellent, well done. So there's a lot. You are really doing very well with this. Excellent. So it's gone. We've got the change in the environment, which is the stimulus. It's being picked up by the receptors, passed on the sensory neuron to the relay neuron to the motor neuron, and then that goes to the effector, which carries out the response. And in between the neurons are these synapses, these little gaps where the impulse has to be passed along. Excellent. Okay, so, and don't forget the muscle or gland is the effector. So it's a muscle or a gland is the effector. So it could be you've picked up something really hot and then you drop it. So the effector is the muscle in your hand because you've dropped it. Or like with the sweating, it could be a gland, a sweat gland, to make you sweat more if you're getting hot. Okay, good. Right, well, let's move on then and actually look at synapses, shall we? So let's have a look. What connects neurons? How does the signal pass from one neuron to the next? So if neurons are separate cells, how do electrical impulses travel between them? So the electrical signal must be passed along somehow. So here's the end of one neuron, okay, and here's another neuron, and they're going to join on to another neuron. So these might be sensory neurons going to a relay neuron. How are they going to pass from here? So they've come along this way, and we want them to go across and somehow communicate with the next neuron. So how's this going to happen? Brilliant. Neurotransmitters, that is correct. So we have something called neurotransmitter, don't we? A lot of the words in biology are really good because neuro obviously mean nervous, and transmitter, it's passing it on, isn't it? It's transmitting it, it's carrying it across by diffusion. Excellent, brilliant. Okay, converted into chemicals. So that electrical signal must be converted into a chemical signal to move between the neurons. So the chemical signal is called a neurotransmitter. Neuro for neuron, transmitter as it transmits or carries it across the gap. And this is another example of diffusion. So basically, these synapses are going to release neurotransmitter, which then diffuses across the gap. So we'll have a look at these in a little bit more detail. All right. So as you notice, it's not just normally one neuron joining onto one other. It's a network, a whole network of neurons across the body, around the body. And there may be lots of them coming into one, or one branching off into lots. So it's a bit like a series of roads around the body. So you must remember, it's not just one neuron joining onto another neuron. It's several neurons coming in to connect with one other, say. All right. Let's have a look then. Let's have a look at a little bit more detail of the synapses then. Okay. So here is a synapse. So the gap is the synapse. And let's say the impulse is coming in here, isn't it? All right. What direction? How does it only move in one direction? All right, we need that impulse just to move in one direction, from the receptor all the way through, down the sensory neuron to the relay neuron to the motor neuron to the effector. We don't want it bouncing backwards and forwards because it's going to cause an awful lot of confusion. So how do we make sure it only goes in one direction? Well, we'll have to have a look at this. Have a look at the, here's the axon, the end of the axon, and this is the presynaptic bulb or knob, you can call it. So this is a presynaptic bulb. All right. What's here in these little vesicles? Does anybody know what those vesicles contain? We've talked about them already. But these vesicles are very important, and they make sure that the information gets passed along. So what is it that's in these vesicles? Any idea? Yay, we've got the neurotransmitter. So we've got neurotransmitter. And they are in these vesicles. So what happens to these vesicles? Brilliant. Well done, Daniela and Ash. So how does this neurotransmitter diffuse across a gap? At the moment, where I'm showing here, it's just in a vesicle at the moment, isn't it? What's going to happen to it? Any idea what happens to these vesicles? Clue is obviously in the diagram. What do you think happens? So what do you think happens to these vesicles? Are they just going to sit there in the presynaptic bulb? Ah, you're close to that. Yeah, you're getting endocytosis and exocytosis confused, but actually those are A-level terms. So you're doing really, oh my goodness me, we're using some really good A-level stuff here. Calcium causes it to move to the presynaptic membrane. You're absolutely right. Yes, exocytosis. Well done. So those are words which are brilliant words, and they're used a lot in A-level biology. They're not always used in GCSE. No, all the school exams are not the neurotransmitters are not all the same. All right. So the terms are not all the same. No, the school exams are not all the same. And we're using quite technical terms there. All right. So Right. So what we need to do is, all we need to do is say that the neurotransmitters are released, and you can say by exocytosis, and they diffuse across the synapse. Sorry, my watch was answering me, so I just had to put it on silence. There we go. And diffuse across the synapse. So here they are, they've been released into the gap. There we go. And they're going to diffuse across that gap, aren't they? All right. So what's going to happen next? Number 2, we've got them diffusing across the synapse. What is happening in this part, number 2? What have they joined onto? Have you got any idea what those neurotransmitters have joined onto? Have you got any idea what these blue things are representing? So the neurotransmitter has diffused across the synapse. Yay, brilliant. And they bind to receptors. Well done, Ash. Yeah, so it's like an active site. It works like an active site because they are they match together so that the neurotransmitter fits into the receptor, like jigsaw pieces joining together. All right. Just like active sites on enzymes. So neurotransmitter binds to receptors, and that's on the postsynaptic membrane. And that triggers another response. So it passes the impulse from one neuron to the next. And so this, on this side, it's the axon, and on this side, we've got the dendrite, which has the postsynaptic membrane. So on that postsynaptic membrane are these receptors, all right? And what happens, they're complementary in shape to the neurotransmitter. So the neurotransmitter binds onto them like jigsaw puzzles joining together. And then what happens is the impulse is passed on. Now the impulse can only move in one direction because the neurotransmitter is only released on one side, the presynaptic side, and there's only receptors on the postsynaptic side. So that ensures that the impulse only travels in one direction. Okay. Now, we don't want that impulse constantly being sent. And if that neurotransmitter is bound to a receptor, it's going to keep on triggering a response. So what is going to happen at the end? They do look a little bit like enzymes, and that's what's coming up next, because we don't want those neurotransmitters bound to those receptors for the whole time. So if we got any idea what happens to those receptors, or sorry, the neurotransmitter bound to the receptors. And the last comment does help with this. So we need to get rid of that neurotransmitter off those receptors. Brilliant, they're broken down by enzymes. So enzymes break down the neurotransmitters. So once that impulse has been sent on, then the enzymes break down the neurotransmitters to stop multiple signals. Brilliant. So we've got the impulse has arrived. It's triggered the neurotransmitters to be released from the vesicles, because the vesicles move towards the presynaptic membrane. They release the neurotransmitter into the synaptic gap, and then they move across the synaptic gap by diffusion. They bind onto the receptor, and then that triggers the impulse in the next neuron. And then enzymes break them down, so we haven't got this constant passing on of impulses. Right, increases the speed of impulse. The concentration of neurotransmitter determines how quickly it diffuses across. And if there isn't enough release, because there isn't enough of a stimulus, then it won't get across. All right. but it will allow for the next impulse to be sent, won't it? So that's brilliant. Okay, so how confident are you feeling with this topic? Are we ready for some exam questions? So do you think it's quite straightforward, number 1, all right, 2, or super confused, number 3? What do we reckon? 1, brilliant, excellent. Yeah, well, this is a good chance to just check you out and see whether you're happy with this, because we've got some exam questions coming up. All right, so let's have a look at this first one then. This is from AQA. All right, so figure 3 shows how a nerve impulse passing along a relay neuron causes an impulse to be sent along another type of neuron, neuron X. So we have got a relay neuron coming in. So what type of neuron does the relay neuron pass on? Okay, wow, Jennifer, you are definitely looking at A-level standard again, acetylcholine and cholinergic synapses. Yes, it's a motor neuron. The next one is a motor neuron, because we've got that SR, haven't we? Sensory neuron, relay neuron, motor neuron. Fantastic, well done. And then describe how information passes from the relay neuron to neuron X. So we've got some of this about, yeah, so we've got, it is actually second year biology, A-level biology, the acetylcholine and cholinergic synapses, that's true. So let's stick to the GCSE that we've just talked about. Describe how information passes from the relay neuron to the motor neuron. Neurotransmitters are released. So the relay neuron releases neurotransmitters. So that's 1 mark, but we need 3 marks, don't we? So what happens with the neurotransmitter? Neurotransmitter releases calcium ions to disrupt vesicles containing the neurotransmitter. The nerve, which is good, yeah. The nerve impulse from relay is carried across, yeah, brilliant. So the neurotransmitter is then going to diffuse across, isn't it? Yeah, so you can call it synaptic gap or synaptic cleft. It really doesn't matter. Both of those are correct. And it attaches, yeah. Brilliant. The neurotransmitter is broken down by enzymes and they join to receptors. Good. So before they're broken down by the enzymes, they're going to attach to receptors on postsynaptic membrane of motor neuron. And that, of course, then passes on the impulse, doesn't it? Well done. There's some really good answers here. Right, so let's have a look. We've got another one coming up. Let's have a look at this one. So scientists investigated the effect of 2 toxins on the way in which information passes across synapses. So we've got curare and strychnine. So curare decreases the effect of the chemical on the motor neuron, and strychnine increases the amount of the chemical made in the relay neuron. Describe the effect of each of the toxins on the response by muscles. So let's have a look at the curare first then. It decreases the effect of the chemical on the neuron. So what's it going to do? What's it going to do to the muscles? Is it going to make them contract more or contract less? What do you reckon? You can put more or less. What do you reckon? That's a nice easy one to write down, isn't it? Yeah, less, brilliant. Decreases, well done. Decreases contractions of the muscle. So it decreases the contractions of the muscle because it's decreasing the effect on the neuron X. Brilliant. So what about strychnine? What's strychnine going to do? Because this is increasing the amount of the chemical, the neurotransmitter made in the relay neuron. So what do we reckon is going to happen to the contractions of the muscle when strychnine is used? More contractions. Excellent. Causes more contractions. Well done. Brilliant. Well done. Really good answers. Well, I think you seem to be able to cope with that one quite well. So let's have a look at the next part of the class then. We're going to have a look at reaction times and reflexes. All right. So, all right. So let's have a look at reaction times and reflexes. So we need to have quick reactions, don't we, to defend our body from harm, all right? So what are reflexes? Does anybody know what a reflex is? So we need to respond rapidly to danger in order to, what do we reckon? What are reflexes? What's the point of reflexes? How fast we yes, brilliant. So we're responding to a stimulus, isn't it? So yeah, it's an action caused by a stimulus, immediate response to a stimulus. Brilliant. Immediate is very important. They are rapid reactions, which are involuntary. We don't actually have to think about them, which is fantastic. So we don't have to think about these responses. So the whole purpose of them, like I said, yeah, involuntary response to external stimuli, unconscious. For survival, excellent. So they're to protect the body from immediate danger. Good, well done. Prevent damage. And like you said, they're unconscious, so they bypass conscious decision-making. Okay, so they're rapid, like you say, and involuntary. So they're bypassing the brain. So they're bypassing conscious decision-making. Now, obviously, some of them do go through the brain because things like blinking of the eye go through the brain because that's the closest part of the central nervous system. But you don't have to consciously think about blinking, do you? Yeah, so it goes basically, it goes through the nearest part of the central nervous system. So if you tread on a drawing pin, all right, you're going to pull your foot up from that drawing pin very rapidly. And what's happened is your reflex has gone through the base of your spine, right at the very bottom of your spinal cord. All right. If you pick up something hot with your hand, it's going to go through the central nervous system, which is in line with your arm. If something's coming rapidly towards your eye, say a fly is coming towards your eye, you are going to blink, and that's going to go through the brain. So basically a reflex action goes through the nearest part of the central nervous system and brings about this involuntary response. Okay. So, and it's just basically what you're born with. Those nervous connections are all they are just naturally connected. That is how your nervous system is put together because it has to protect your body. And babies have reflexes, don't they? All right. So we are born with these nervous neural pathways already embedded into our nervous system. So we don't have to think about it, and it's all there as a survival, because if we didn't have reflexes, we would die very early on in our life. So if you look at babies, they put their arms out if they think they're going to fall, and that's to protect their body. So we're born with them. Is that okay? Hopefully that makes sense. Yeah, when we touch a sharp object, obviously we will withdraw our hand, won't we? So that will go through the nearest part of the central nervous system to our hand, won't it? All right. So those are all examples. So this one is obviously dust is blowing towards the eye, and the eye is going to just automatically blink to protect the eye from getting scratched, because if the eye gets scratched, it's going to damage your eyesight. So we end up blinking, using our eyelids to blink. The muscle is in the eyelid. You blink your eye, you stop the dust from coming towards you, and it's a rapid involuntary reflex. Now, it has gone through the brain because that's the nearest part of the central nervous system, but you haven't had to think about it. It's bypassed conscious decision making. All right, you're aware that you have blinked afterwards, but you don't have to think about it. All right, any questions about that? Does that make sense about what reflexes are? All right, good. So some of you need to know about a classic one for your exam boards are reaction times. Now, has anybody done any reaction time experiments at school? I do this one with the ruler and the dropping of the ruler. It's quite a simple one then, isn't it? Very quick. So now let's look at how we can investigate these reaction times. So it's hard to truly measure the reflex reaction time, as knowing the test is happening will obviously introduce conscious decision making, but we can investigate overall reaction time by investigating here the speed of catching a falling object. Yeah? Yeah, you remember doing the ruler one in class. Sometimes people use computer simulations as well. And there's one I've seen on television where they do the touching of the lights, and when a light comes on, they have to touch the light. And that's also a good one for testing your reaction time. But at school, when you're in class, the classic one that we do is this ruler one. All right, and it has come up in exams as well. So it's the speed of catching a falling object. So here it's a ruler. All right. So we have to think about the variables in any experiment that we're going to be doing. All right. So these normally what happens is you work in pairs, and one person holds the ruler above the test subject's hand, and then they suddenly drop the ruler, and the test subject then tries to catch the ruler as quickly as possible. And once caught, you can measure the distance the ruler dropped through the hand by recording what number the hand is on the ruler. And then obviously you're going to have to repeat it, because repeats make it more repeatable, more reliable. All right. I will go through these variables with you. So I'm just quickly explaining what the experiment involves, and then we'll talk about how we do these variables and the importance of the variables and how to remember which one's which, because that's incredibly important and people forget the different types of variables. All right. So you're going to be repeating the test to calculate a mean. Obviously, you have to be careful to make sure the test subject always place their hand in the same starting position, and the person dropping the ruler always drops the ruler from the same height to make sure it is a fair and valid test. And then once you have the results, you can then convert the distance to how rapid somebody's response is. All right. So we're talking about catching a ruler and finding out who's got the fastest reaction time. So when we're thinking, normally what happens with this is I do it looking at how reaction times are affected by noise in the classroom. All right. So if you were looking at comparing, let's take it that we do it when everybody's in silence, and then we do it when there's lots of noise in the classroom. We've now got to think about what the independent and dependent variable is. All right. Now, the independent variable is what you choose to change. So it's what you're investigating, what you choose to change or investigate. All right. So you're investigating the independent variable. That's the way to remember it. All right. So if I was talking about noise in the class, that would be my independent variable. I would be changing it from silent to noisy. All right, so that's known as distraction. Anybody done any other ones? Anything else that could affect your reaction time? Any idea of any others that we could have? Yeah, brilliant. The control, we will get onto the controls. All right. All right, so it could be distraction, or we could be looking at caffeine, or we could look at age, because I know that reaction times change with age. So we're looking at what's affecting the reaction time. So the dependent variable is what you measure. And the dependent variable, what you measure, depends on what you're changing. All right. Brilliant. A stimulant. Yeah, stimulants such as caffeine. All right. So that's really good. Fan in the room. Yeah. Drugs. Oh, yeah, that would have an effect, wouldn't it? All right. So there's lots of things. Now, obviously, you can't do about using drugs in a classroom. But if you were going to be out there and checking people who do take drugs, you could actually look at how their reaction rate changes, because that's why certain drugs are banned for people who are driving. So even certain painkillers are banned from using if you're driving, because it affects your reaction time, and therefore you're more likely to have an accident. All right. So distraction, caffeine, age, stimulant, all of those things could affect your reaction time. So those are your independent variable. It's what you're investigating. So you're investigating how these things change the reaction time. So what are we measuring here? In this experiment, what are we measuring? This is the dependent variable, what we are measuring. So having a look at this picture here, what are we measuring in this picture? The length of the ruler is caught at. Excellent. So yeah, brilliant. Time taken to catch a ruler. Well, it's actually, yes, because the time taken to catch a ruler, the longer they take, the further down the ruler it is that they catch it, isn't it? So it's the catch distance in centimeters. All right. So the dependent variable depends on the independent variable. So you would expect if there was a distraction that the catch distance is longer, because it takes them longer to respond, because they might be distracted by what people are saying around them. All right. And then we have the controlled variables. All right, the controls. This is something that we have to keep the same. What do we keep the same to make it a fair test? So what do we have to keep the same? I can see somebody's already written the ruler length. Good. Yeah, ruler length. And then maybe gender. You might want to keep the same. Where the person is dropping it from. So where it's being dropped from. And also the position of the arm of the catcher. Body temperature. Yeah, the temperature of the room could affect it, couldn't it? Because if it's colder, I know I wouldn't be able to catch it as well. So yeah, that's good. Age, yes, because if you're not investigating age in the independent variable, then yes, you could keep them all the same age as well. So it could be age and gender. All right, fantastic. Now, obviously, if you were going to plot this on a graph, just so that those people who are asking about variables, the independent variable is the basis of an experiment. It's what you've chosen to change. So it's the base of an experiment. So that always goes on the x-axis, because that's the base of the graph. And the dependent variable is what you're finding out. So it goes up the y-axis. So those are all the things about variables that you need to know. Are there any questions about variables there? Are you all happy with what we've talked about in this experiment? Yeah? Are there any questions before we move on? Right, let's have a look then. The reflex arc. This is the next bit that we need to look at, is the reflex arc. So remembering back to SRSRMER, we start with the stimulus. So here, what is the stimulus? The stimulus is a change in the environment, by the way. So here, what is it that is the stimulus? Can anybody write down what the stimulus is here? Looking at the picture. Just wait for you. Pathway for the conduction of the nerve impulse. The hot object, yeah. So the heat of the candle, the high temperature, good. So we've got heat, or it could be pain, couldn't it? If you wanted to, pain, heat, caused by the candle. And that's what brings about, so you're right, the pathway for the conduction of the nerve. So the stimulus is what triggers the response, isn't it? Yeah. Yes, brilliant. Detected by thermoreceptors. We talked about that right at the beginning of the web class, didn't we? So after the stimulus, we get receptors, don't we? All right. Receptors. What comes after that? Remembering SRSRMER. So we've had stimulus, receptor. What comes after receptor? Can anybody remember? What's the S after receptor? It's number 2. Sensory neuron, brilliant. Well done, everybody. Sensory neuron. So you can imagine what my next question is going to be for number 3. So we've got the sensory neuron. What's number 3 then? So we've gone SRS. What's the next? Brilliant. Well done. Yeah, that's the relay neuron, which is found in the spinal cord, isn't it? Brilliant. So we've got SRSR. What comes after that then? What's the M for number 4? Associative neuron. Yeah, that's one that isn't used very often. Brilliant. The motor neuron. So that's the M. And what comes after the motor neuron? It's the E. What's the E? Number 5. Brilliant. The effector, which here is the muscle, isn't it? So here it's a muscle. And then what's going to be the response? Because we've done the effector, now we're on the response. What is going to be the response? What's going to happen? What's that effector going to do? Ah, brilliant. The arm is, yeah, pull the handle away. So we have muscle, contraction, withdrawal of hand from the candle. Yeah, brilliant. Fantastic. Well done. Well, you seem to be doing really well with this. So let's have a look at how confident you're feeling so that we can have a look at some exam questions. How are we feeling? 1, 2, or 3? Are we all happy? Yeah, 1. Brilliant. Well done. Let's have a look at this one. Going back to the start of reflexes, explain the advantage of the reflex arc. Brilliant. I like that one, of course. So explain the advantage of a reflex arc. And we're looking for 2 marks here. So we need to give 2 points to this. What do we think? Explain the advantage of the reflex arc. There was 2 main things we talked about when we talked about the importance of reflexes. What were they for? First of all, how quick are they? Helps prevent damage. Excellent. Okay. Protect us. Fast. Prevented. Yes. Brilliant. So reflexes allow a rapid, brilliant, unconscious response, isn't it? To limit damage from danger. Yeah, unintentionally, we call it, it's an unconscious thought. We haven't had to put it through the conscious brain. doesn't go through conscious thought. Automatic, involuntary is a good way of putting it. Automatic, involuntary, immediate and quick. Those are brilliant A-level result answers as well. Fantastic. Right, let's have a look at this one then. A little bit of maths in here. The student did each measurement 3 times to calculate a mean value. Well done, student. So we always have to do repeats. The reaction times for the 30-year-old person was 192, 174 and 180 milliseconds. Calculate the mean reaction time of the 30-year-old person. So quickly write down what you think the answer is to that. And then we'll have a look at the tick boxes and see whether you've got which one you're going to go for, whether you're going to go for 1, 2, or 3. Right, so how are we going to do with the second question? Have a thing, add them up and divide by 3. Brilliant. So it's 192 plus 174 plus 180 divided by 3, which gives us 182. Excellent. And which one of the following is an advantage of repeating each test 3 times and not doing the test just once? Brilliant. Well done. It's not actually the second one. Ah, that's interesting. It's the measurement that would make it more precise, not doing repeats. So how you measure it, if you had a more accurate way of measuring it, that would make the results more precise. Yeah, so we've got to be really careful. The instruments you use will make it more precise, but we need to identify any anomalies. All right, we can't say there's not going to be any errors, so we can't do 3 because we can't say that there's going to be no errors. All we can do is identify any errors or anomalies. So it's actually 1. That's a really good question. People think tick boxes are easy, but they're not always easy, are they? All right. So quickly, the last question, because I then want to show you about doing the tutoring and seeing how you can get on with it. All right, so the last one. Some people think that old people should not be allowed to drive a car. Why is it more dangerous for old people to drive cars? Use the information from table 1 to support your answer. Okay, so what has happened to the reaction time as we've gone older? Look at 90-year-olds. What's happened? Reaction times, yeah, much longer, are too slow. That's why people say, and that's why you have to redo your test if you want to drive a large vehicle. Yeah, so they are more likely to have an accident. Yeah, brilliant. Well done. Okay, so we've had a good look at the synapses, communication between neurons. We've described the structure and function of a reflex arc, and we've described the importance of reflexes in the investigation of reaction times. Brilliant. So what I want to just talk to you about is, obviously there's upcoming web classes, and we've got maths, physics and chemistry coming up, all live on, we've got them coming up on YouTube. And of course, I need to remind you about the fact that you can get a week free. Why not? You have nothing to lose. And with this, you're going to get 4 or more classes per week. You get a full curriculum is going to be covered. Obviously, you're going to get handouts as well, and you can re-look over previous classes. So it means that you've basically got the teacher there all the time for all of those topics. And so it's really useful. If you don't understand something, you can go back over it by clicking on past recordings, and you can get one-to-one support. All right. And to do that, you just need to enter your code at the checkout. All right. at SnapRevise. So what I want to do now is I want to try to just take you over so you can see how to do this. So I am just going to hopefully take us into the Google Chrome. So let's have a look. And what I want to do now is I just want to quickly show you okay, so here we have the free trial, and you just click on this start the free trial and you enter the code in, and then you click at the tutoring at the top, and the tutoring at the top then shows you all the different topics. So you've got A-level biology, chemistry and maths and physics, and then you've got GCSE chemistry, GCSE biology, maths and physics. And then you just click on whichever you want and you can click join now. All right. And with that, you can then enter your code. All right. And then you've got all of the things about, you can go to your different areas of it, and at the bottom there is, you can have a look at an example, and then of course here is the what's coming up, so you can see exactly what topic is coming up and when. All right, so it's well worth doing, and what have you got to lose if you've got 1 week free, is what I say. I always like to take the idea of just checking out anything that is available to help you. All right. So hopefully that all made sense. Has anybody got any questions for me before we come to the end of the lesson? Does anybody want to ask me anything about the lesson today? Okay. Well, it was so good to see so many of you here from so many different countries, which is brilliant. And I know that lots of people are doing different exam boards, but we make sure that we cover what is needed for each of the exam boards. All right. So hopefully I'll see you again. I will be doing my web class again tomorrow. Lovely. Thank you, Zara. I'll be doing my web class tomorrow as usual, and Wednesday, and drop in on Thursday. Drop in is brilliant. You can always ask to go over particular topics. I'm really glad you've enjoyed it. That's fantastic. All right. So take care, and I will see you all, I hope, again very soon.